Hello, I'm Dr. Tim Jansen. I'm a family practice doctor in Portland, Oregon, who has a major interest in genetic genealogy, particularly in relationship to autosomal DNA. And today I'm going to be talking to you about how you can trace your ancestral lines in the 1700s using DNA, particularly autosomal DNA. As some background, what I'm going to do is I'm going to provide some additional information about autosomal DNA and how you can use it for genealogical purposes. I'm also going to talk about your DNA matches and how to best organize them. And I'm also going to give you some background about doing autosomal DNA analysis. The first thing I'd like to talk about is the structure of a cell and where we get our DNA. Our bodies are made up of trillions of cells, and these cells all have a nucleus inside of them, which has our autosomal DNA and also our X chromosome and our Y chromosomes, depending on our sex. In addition, there's also the mitochondria, which also have their separate genomes, 16,000 base pairs in the mitochondria, and those little organelles provide additional background because the mitochondria are passed down through the maternal line of descent. The X and the Y chromosomes are passed down from the male and the female, depending on the inheritance pattern. Our autosomal DNA is passed down from both parents. Here's a karyotype. What this shows is all of the different types of chromosomes that we have in our genome, starting with chromosome 1, and then continuing down to chromosome 22. The longest chromosomes are given the first numbers, and then the shortest chromosomes are 21 and 22 with the highest numbers. In addition, you can also see the X chromosome and the Y chromosome. This happens to be a male, so you can see one X chromosome and one Y chromosome. If this was a female, you would see two X chromosomes. Autosomal DNA is used to determine how people are related to each other on any line of descent if they share at least one ancestor in common in the relatively recent past. So autosomal DNA has a lot of potential for genealogical researchers because there's a total of 44 autosomal chromosomes. There are about 1 billion base pairs in all of these chromosomes. So it's a lot of genetic material and a lot of data. Unfortunately, the analysis of this data is somewhat complicated by the fact that each parent passes down only about half of their autosomal DNA to any one specific child. So there's constant dilution of the DNA as it's passed from one generation to another. Autosomal DNA is of most benefit in determining relationships between people who share a common ancestor in the last 6 to 8 generations, and it's particularly helpful in the last 4 to 5 generations. 6 to 8 generations can still be used if you use caution in your analysis. If you're going to do DNA testing, there are a number of different DNA testing labs that cater to genetic genealogists you might want to test with. I've listed the 5 most commonly chosen DNA testing labs on this particular slide: 23andMe, Family Tree DNA's Family Finder test, and Ancestry.com's DNA test, as well as MyHeritage's DNA test, and finally Living DNA. If you want to learn more about the various different testing companies, I would suggest you take a look at the ISOG Wiki chart that I've created that compares and contrasts the various different DNA tests offered by each of these testing labs. So let's ponder the question, why should genealogists want to do autosomal DNA testing in the first place? First of all, autosomal DNA testing is a wonderfully powerful tool to help you verify the accuracy of your family tree. When you start off doing genealogical research, and particularly if you haven't done any autosomal DNA testing, you don't really know for sure if your family tree is accurate. And so DNA testing allows you to create a means to determine if you've actually researched the correct biological ancestors that you think you have. Autosomal DNA testing is also very helpful if you've got some genealogical brick walls in the early 1800s or even into the 1700s that exist because you don't have adequate genealogical records for those particular ancestors. So autosomal DNA is simply another means by which getting around genealogical roadblocks. Another interesting thing you can learn from autosomal DNA testing is insights into your ethnic background or what regions of the country or the world that you came from. So, for instance, my father happens to have a story of some Ashkenazi Jewish ancestry among his many Mennonite ancestral lines. And so when I did his autosomal DNA test, it did indeed confirm that he had Ashkenazi Jewish ancestry. And similarly, my mother has a history that she descends from Chief Tarhi, who was a Wyandot Indian. And the autosomal DNA test that she has done has also verified that she has Native American ancestry. So let's talk about some of the basics about autosomal DNA testing. First of all, you need to know that there are around 7,000 centimorgans in the human genome. And this is the genetic data that we use for all of the comparisons that we are trying to do when we do autosomal DNA testing. As a general rule, we don't look at matches much less than about 7 centimorgans. You can sometimes go down to 2 to 3 centimorgans in selected situations, but in most people's cases, they're looking at matches over 7 centimorgans. So there's about 1,000 or so different triangulated groups that you can utilize out of your 7,000 centimorgans that you're carrying in your autosomal DNA. Another point to be made is that generally speaking, all ancestors contribute at least a portion of their autosomal DNA to their descendants going back at least 5 to 6 generations. However, as you start going back further in time, your genetic ancestors are not necessarily the same as your genealogical ancestors. So if you go back to around 10 generations or so, it's been estimated that only around 12% or so of your ancestors are contributing to your genetic makeup. So in other words, only about 120 or so of your 1,024 ancestors at 10 generations back are actually your genetic ancestors. Here's some other basics that you want to keep in mind. Parents and children share about 50% of their autosomal DNA. Siblings are sharing about 50% of their autosomal DNA. And when you get out to more distant relationships like first cousins, they're averagely sharing about 12.5% of their autosomal DNA. Finally, when you get out to sixth cousins, there's relatively little shared DNA in most cases. However, in some cases, there can be some fairly large segments of DNA that are shared even among sixth cousins. Here's some additional basics in centimorgans. So if we assume that there are 7,000 autosomal centimorgans, then parents and children are going to share about 3,500 centimorgans on average. Siblings are sharing about 2,600 centimorgans on average, of which some of that is half identical and some of that is fully identical. First cousins are sharing about 885 or so centimorgans. Fourth cousins are sharing only about 14 centimorgans on average. I suggest you try to keep these numbers in mind. I suggest you remember that first cousins share about 880 centimorgans, second cousins 220, third cousins 55, and fourth cousins 14. If you can keep those numbers in mind as you look at your matches, it'll be helpful for you. Another important thing to keep in mind is the fact that the amount of shared DNA at the more distant relationships is quite variable. In fact, if you look even just at what first cousins share in common, you can see that there are some first cousins who have been discovered who share only 83 centimorgans and some that share as much as 1,500 centimorgans. When you get to the second cousin once removed level of relationship, you can see that it's possible that there will be no shared DNA between second cousins once removed, but you could share as much as 466 centimorgans with a second cousin once removed. Finally, when you get out to more distant relationships, such as fourth cousins, you can share as much as 55 centimorgans or possibly even more in some cases. And of course, you can share nothing with a fourth cousin as well. The odds of sharing DNA with any specific relative get less and less the more distant the genealogical relationship. So if you're looking at second cousins, as I mentioned in the last slide, there's basically a 100% chance that you're going to share DNA with a known second cousin. However, when you get to third cousins, only about 90% of all third cousins are sharing autosomal DNA segments. And when you get to fourth cousins, about 50% of them are sharing autosomal DNA. Finally, when you get out to the sixth cousin level of relationship, it's relatively uncommon for a sixth cousin to share autosomal DNA with another sixth cousin. In fact, only about 1 or 2% of sixth cousins actually do share autosomal DNA. I want to introduce two basic types of autosomal DNA analysis that you're going to use in your genealogical research. The first is what I refer to as quantitative analysis. This is where one of the people is comparing their DNA to another person, or maybe even more people, to see how much DNA that they share in common with each other. You're trying to see if it meets a certain specific expected amount. And this particular technique can definitely be used back to 4 to 5 generations and possibly back to 6 generations. What you're doing is you're looking to see if the average amount of shared DNA among the people you're comparing is approximately the same amount that you would expect from some of the averages in the previous slides. Another type of analysis is what we refer to as triangulation. So with triangulation, what you're doing is you're reviewing the pedigree charts of people who share the same segment to see if they can find a common ancestor. And you can use this particular technique on all of your triangulated groups, and it can be used multiple generations. So it can go back as much as 10 generations or more, depending on how far the genealogical data is available for any specific group of people. I use both of these techniques together, and in particular, when I'm trying to confirm genealogical relationships 5 to 6 generations back or further. When you're trying to verify the known genealogical relationships, you first want to look at your first and second cousins to say if they share the appropriate amount of DNA with you. Because in some cases, there will be non-parental events that have occurred either through illegitimate births or adoptions that result in an unexpected amount of shared DNA. And you can pick this up, particularly at the first cousin level of relationship or more closely. At second cousins, it sometimes is a little more challenging to spot, but you should be able to see that if you get enough second cousins tested. You're trying to verify that your biologic family tree is the same as the genealogical family tree that you have been researching for all of these years. The other thing you want to do is you want to create clusters of cousins, if at all possible, to verify that the relationships more distant than second cousin are actually true. So you want to verify third cousin relationships, fourth cousin relationships, and if possible, fifth cousin genealogical relationships. So you want to look back at some of the statistics that I've pointed out previously at the ISOG wiki in the autosomal DNA statistics section to look at the expected amounts of DNA among all of these people. Another important thing for you to do is to maintain a match list of all your close relatives who's tested. As a general rule, it's a good idea to maintain an Excel file for all your known genealogical relatives out to the fifth cousin level of relationship. And I include lots of different type of information in this file, including their names and contact information, etc., as I've included on this particular slide. Another type of file that I maintain is what I refer to as a cousin clusters file. And when I do this type of file, what I do is I include the names of all of the relatives who've been tested on any specific ancestral line. And this allows me to quickly look at my contacts and my matches in a specific ancestral line if I'm going to do additional research or analysis on that particular line. And I have these organized by family tree, just like they would be in my pedigree charts. And I've hidden the names underneath, but in my Excel file, I can look at all the names in any one particular ancestral line. There's quite a few different sources you can use for finding cousins to run your genetic comparisons against. And I've listed these here before, and I'll mention GEDmatch, which is a independent company with over 1 million people in its database that you can also run comparisons at. Wikitree is a great source for you to find additional cousins that you hadn't been aware of. And in the next presentation, I'll be using some of the people included on this particular slide. And you can look up any ancestor of yours and see who's been tested and potentially get their GEDmatch kit number. Another thing that I do is I look at tag groups at GEDmatch. So I've created various different tag groups, and I can readily run comparisons among these various different tag groups. And it allows me very quickly to compare one new match from a specific ancestral line against everybody else in that particular ancestral line relatively quickly. So let's summarize the key points in quantitative autosomal DNA analysis. First of all, you're going to target your oldest generation for testing. They have the most DNA, and so you're going to try to maximize the amount of DNA that you capture by testing them first. Also, maximize the number of people you test, particularly on various different ancestral lines. And then analyze the people who should be most closely related to each other first. So their analysis is generally going to be more accurate than more distantly related clusters. Also, make sure that you do separate analyses for each match at each generational level. So if you're looking at second cousins, run all the second cousins first, and then second cousins once removed, third cousins, etc. Also, make sure that you discard any half-identical regions that are passed down through a different ancestral line than the one that you're analyzing. And you have to do chromosome mapping in order to sort this out. Be very careful also with any half-identical regions under 5 centimorgans, because these are frequently false matches. And finally, always be willing to review your previous conclusions that you may have drawn about genealogical relationships once you have new data that comes in. So let's talk about some strategies for breaking through your genealogical brick walls. So first of all, you're going to create clusters of cousins you descend from each of the ancestors who are descended from your genealogical brick walls. You want to map the chromosomes of each of the people in these clusters as best as you possibly can and maintain a master matchless spreadsheet for all your older relatives. You keep all this matchless spreadsheet organized as best as you can into triangulated groups. Then you want to study the pedigree charts of all your matches on any particular segment who are linked to an ancestor who's in your genealogical brick wall. So you want to get all the descendants who've been tested from that particular ancestor and have their data ready for review. And particularly, you want to review the pedigree charts of people who share the most centimorgans on average with your cluster of relatives in that particular line of descent. You also want to pay for additional people to be tested if possible. And if possible, get Y and mitochondrial DNA sequences for the ancestral lines that you're researching. So you have multiple different ancestral lines you might want to get both Y chromosome and mitochondrial DNA sequences for. I also encourage people to upload to GEDmatch because GEDmatch provides the greatest flexibility in terms of running your comparisons. Finally, I want to conclude with a summary of my mother's pedigree chart, which I'm including here. And it shows all the ancestral lines that I verified using autosomal DNA testing. My great-grandfather, Charles Youngman, he has some holes in his family tree yet, and hopefully I will be able to fill those in, but I verified all of his other lines as outlined here in green. However, you can see on the next ancestral line for my great-grandmother, Anna Brown Armstrong, that some of her lines have not yet been verified using DNA testing, and those are depicted in black, such as Jeremiah, Brown, and Hannah. I have other ancestral lines similar to this, and you can see where my brick walls are that I'm still continuing to research even today. If you have questions, feel free to contact me at my email address, tjansen@comcast.net. Thank you very much for listening to this presentation. I'll continue with the second presentation next on a specific research problem on my Meeker line. That'll be in part 2.